Thank you for inviting me to present my project that I've been working on as a postdoctoral fellow in John Digg's lab. The project I'm going to talk to you about focuses on genetics that underlie leukemic transformation and myeloproliferative neoplasms. So what are myeloproliferative neoplasms, or MPNs? These are chronic myeloid malignancies that are characterized by the proliferation of one or more of the following myeloid lineages, so granulocytic, erythroid, or megakaryocytic. The 2016 WHO classification of MPNs it includes a heterogeneous group of diseases, which can be divided into those that are Philadelphia positive, and this includes chronic myeloid leukemia, which is owing to the presence of the Philadelphia chromosome that leads to the BCR-ABL translocation. And the remainder of the MPNs are considered Philadelphia negative, and amongst these, polycythemia vera, essential thrombocythemia, and primary myelofibrosis are more common ones. So polycythemia vera is characterized by the proliferation of erythroid lineage, which leads to increase in red blood cell production. ET is characterized by proliferation of the megakaryocytic lineage and overproduction of platelets. In primary myelofibrosis, you do see in the bone marrow, you can see granulocytic or megakaryocytic proliferation. However, this particular entity is characterized by the presence of bone marrow fibrosis, which is absent in ET and PV. So although fibrosis is minimal in PV and ET, the risk of developing fibrosis later on in disease is present. What is important to also note in terms of clinical features of these diseases is that the risk of leukemic transformation for all three is present. So ET has the lowest risk of leukemic transformation. However, this increases when you have primary myelofibrosis. MPNs have three disease phases. The chronic phase is characterized by having less than 10% blasts in the bone marrow. This can progress to an accelerated phase where you have 10 to 19% blasts. And you can proceed further to a blast phase, or what is considered at this point acute leukemia, when you have 20 or more % blasts. As I mentioned earlier, the PMF has the highest risk for transformation to blast phase. However, PV and ET, once these have progressed to fibrosis, also have a very high risk of transforming. So MPN driver mutations have been described in these three diseases, and these include mutations in JAK2, CALR or calreticulin, and MPL. In polycythemia vera, the majority of PV are due to JAK2 V617F mutation, smaller percentage due to mutations in exon 12, and there's a few that have unknown driver mutations. ET and PMF are predominantly driven by JAK2 V617F, next followed by CALR mutation, and then MPL. And then in a subset, about 10% in both cases, there are no driver mutations in any of these genes seen, and these are considered triple negative. So in addition to these driver mutations, other mutations in myeloid leukemia genes have been described. And in this study by Grinfeld and al., where they looked at over 2,000 MPNs, they performed sequencing of 69 myeloid leukemia genes. And in this cohort, it should be noted that about 65% of the patients in this cohort had ET, meaning that the remainder of the patients had PV or MF. So the prevalence of these mutations may be slightly underestimated for MF, which we already know tend to have a higher mutational burden in other myeloid leukemia genes compared to ET. So just bear that in mind. However, the point of this slide is to point the molecular complexity that exists in MPNs. So what you see beyond the driver mutations in DNMT3A, ASXL1, and TET2 are being the highest prevalence, and then you also still see mutations, other epigenetic modifiers, lysosomal proteins, transcription factors, as well as signaling proteins. There's been many studies that look at molecular mutations and their prognostic impact on clinical outcomes. They have shown that calreticulin mutations overall confer a good clinical outcome. However, mutations in ASXL1, SRSF2, IDH1 and 2, and EZH2, and more recently, U2AF1, specifically Q157 mutation, confer poor clinical outcomes, specifically reduced overall survival and increased leukemic transformation. And these mutations have been referred to as high molecular risk gene mutations. So when you look at blast phase samples, you can see that molecular mutations in myeloid leukemia genes are actually more prevalent at this stage of disease. And you can see that the high molecular risk genes are represented at high frequency here. So although there have been lots of studies looking at molecular mutations and MPNs and then its transformation to blast phase, there are still unanswered questions. Certainly, when do mutations in myeloid leukemia genes occur in relation to transformation is unknown. Which hematopoietic stem and progenitor population these mutations occur in is also unknown. And how do all these different mutations cooperate together to promote blast transformation is also not well delineated. In my project, I hope we aim to answer the first two questions, which look at when these genes are acquired in the process of transformation and in which stem and progenitor population. So in order to do this, we have obtained samples from the Princess Margaret Cancer Center Leukemia Bank. And these samples were collected from patients who have myelofibrosis and who have transformed to blast phase in 9 patients and 1 who transformed to accelerated phase. Samples had been collected at 2 time points. Time point 1 is in chronic phase and time point 2 in blast phase or accelerated phase. As you see here, this indicates the specific diagnosis for each patient. And the driver mutation that is responsible for the MPN was predominantly due to JAK2 V617F. 1 patient had MPL, and the patient with accelerated phase had CALR mutation. The time interval between chronic phase sampling and blast phase diagnosis ranged between 1.5 to 6.6 years. And in the 4 patients, we had intervening time points available for our study. So our experimental workflow was as follows. From the blast phase sample, or time point 2, we performed a sort where we collected leukemic blasts. From the chronic phase sample, we collected the MPN clone, which consisted of mature CD33 positive cells and CD34 positive blasts, which were all collected into one population. We also sorted out T cells for reference control. DNA was obtained and submitted for whole genome sequencing in order to identify blast phase variants as well as chronic phase and somatic variants. In addition to this sort, we also performed a second sort where so from the chronic phase sample, we sorted stem and progenitor populations that are listed here, and also sorted out mature cell populations, which included B, T, lymphoid cells, NK cells, and myeloid cells from that were CD33 positive and also from the erythroid lineage. Whole genome amplification was performed on these sorted populations, and from these populations, we performed clonal tracking of blast phase variants using ddPCR and/or targeted sequencing, which is ongoing at present. From the blast phase sample, we also performed a similar sort, but only collected mature populations, which were also subjected to clonal tracking of blast phase variants. Stem and progenitor populations from this sample, from this time point, was not collected owing to the fact that there would be considerable contamination from leukemic blasts and difficulties in later on interpretation of our results. So these are some of the results from our whole genome sequencing. On this slide, I've indicated the total number of exonic and splicing variants that were detected at blast phase. So these are only looking at the 9 blast phase samples that we have. And we see that there was an average of almost 24 exonic and splicing variants that were identified amongst all the samples, with a range of 18 to 40 variants. Amongst all these exonic splicing variants, we found that there were an average of 5.3 myeloid leukemia genes that were actually mutated, with a range of 2 to 8. per sample. On this side, I've summarized all the myeloid leukemia genes that were mutated. This excludes MPN drivers that were mutated in blast phase specifically. So in about half the samples, or half the variants, they were present in at least 2 or more patients. And if you look at the high molecular risk genes, you see that these are predominantly represented in more patients. So SRSF2 and 5, ASXL1 and 4, and IDH1 and 2 and 4 patients. But all of them were represented in our cohort of 10. When you focus just on MPN driver mutations and look at the varying allele frequencies that were present at chronic phase and blast phase, you can see that in pair 2 and 6, the JAK2 mutation was actually no longer present at blast phase diagnosis. So in this particular, in these 2 cases, this indicates that the leukemia was derived from a clone that was distinct from the MPN clone. On this table, which is a pretty busy table here, but it summarizes all the myeloid leukemia gene variants that were identified on the left-hand side, and on each row represents a mutation or gene, myeloid leukemia gene. Each column represents a paired sample, and the first subcolumn represents the chronic phase sample, and the second subcolumn represents the blast phase sample. And they've also indicated the time to blast phase for each pair. If a variant was present, a variant allele frequency is indicated for that particular sample and gene. So you can see in pair 1, there were 8 variants identified in 8 different myeloid leukemia genes, 7 of which were present at the chronic phase, and only 1, which was in IDH1, was undetectable by whole genome sequencing. And if you look at the remainder of all the pair samples, you see that there is, similarly, there is a range between about 2 to 8 total myeloid leukemia genes present at blast phase. However, there was a range between 0 to 4 variants that were not detectable by whole genome sequencing in the chronic phase. So the next step was then to, in our project, was to do clonal tracking of the blast phase variants using a much more sensitive approach, and we used 2 approaches. The first approach was to do droplet digital PCR, and using this technique, we looked specifically at myeloid leukemia gene variants, and in particular, we focused on blast phase specific variants. We're also performing targeted sequencing through OICR, and we are looking, our panel includes variants that target myeloid leukemia genes, as well as blast phase specific non-coding region variants. The goal of using these 2 approaches is to allow us to interrogate the sorted HSPCs and mature cell populations. This will allow us to determine when myeloid leukemia gene variants are acquired during leukemic transformation, and also allows us to determine the cellular origin of blast phase, meaning within which HSC or which HSPC population do these mutations arise. So on this slide, I'm summarizing ddPCR results specifically for pair 5. In this sample, there were 3 blast phase specific mutations that were not detected by whole genome sequencing. There were 2 variants in KRAS and 1 variant in NRAS. We had intervening samples available for this sample, and we see that in our first time point, using ddPCR, we were not able to detect this variant at 3.6 years prior to blast phase diagnosis. However, this variant becomes apparent and is acquired already by 2 years prior to blast phase diagnosis, and is present at very low allele frequencies within the GMP progenitor population. And we see it also in the CD33 positive myeloid population. This variant is present still at very low frequencies at 1.5 years, and we can now see it within the HSC population and downstream progenitor, and again in the myeloid population. As we get closer towards blast phase, and about 6 months prior to diagnosis of leukemia, we see the expansion of this variant. And by the time we're at blast phase diagnosis, it makes up 33%. It has a 3 variant allele frequency in the leukemia blast. So similarly, a KRAS variant, another KRAS variant and the NRAS variant were not detectable at 3.6 years. However, interestingly, both of these variants were acquired actually at 2 years, as evidenced here in the myeloid population here and in the NK population. And what's interesting in the NRAS variant, we also detect this in the HSC population at very low frequencies and in downstream progenitor and mature cells. And similarly, the variant 1 for KRAS. And again, this expands as we get closer to the blast phase diagnosis. And at blast phase, these variants contribute different frequencies to the leukemic blast. So what our results show is that by ddPCR, we are actually able to determine that clones that were fated to transform to leukemia were already present at very low frequencies several years prior to leukemia development, and that acquisition of somatic mutations occurred within an HSC. So on this table, to summarize what I just showed you, we see that in pair 5, these blast phase specific variants were detectable at low frequency, not at 3.6 years, but at 2 years because we had intervening samples. Similarly, in pair 1 and in pairs 8 and 10, we were now able to detect also other blast phase specific variants at very low frequencies using ddPCR. Pair 7, we were not able to generate a primer and probe, a working primer and probe set for ddPCR, and so we're awaiting targeted sequencing results to confirm this particular sample. When we look at pair 2 and pair 6, our ddPCR results confirmed that these blast phase samples were in fact JAK2 negative, and that ddPCR of these blast phase specific variants did not identify these variants at 4.4 years in pair 2, and in pair 6, at 6.2 years and at 1.1 year, we could not identify RUNX1 or STAG2. However, a sample that was available 5 months from diagnosis, we were able to detect high percentages of RUNX1 and STAG2 at 5 months prior to diagnosis. This is in contrast to our other samples, which were blast phase samples that were derived from an MPN clone, and in this case, this was from a non-MPN clone. So furthermore, as I showed you before, in pair 5, these mutations occurred in an HSC. Similarly, in pair 1 and pair 8, we were able to identify a variant in HSC, and this particular pair 8 sample, it was highly suggestive of an HSC as well. So our results so far have shown that the clonal evolution of myelofibrosis can occur in different manners. So we have shown that in pair 1 and pair 10, we see a linear clonal evolution. We can also see branching evolution as evidenced by pair 5 and pair 8. And in our 1 sample, or in 2 samples, we saw clonal evolution the blast phase clone was derived from a distinct MPN, a clone distinct from the MPN clone. So in conclusion, in blast phase disease that's derived from an MPN clone, we see that clones fated to transform to leukemia were present at very low frequencies several years prior to leukemia development. This indicates that mechanisms beyond the acquisition of mutations in myeloid leukemia genes are necessary to effectively promote full leukemic transformation. These other mechanisms may include and are not necessarily restricted to epigenetic alterations, acquisition of non-coding mutations, and inflammation. And we also show that clones fated to progress to blast phase are likely HSCs. We also see that, there's also suggestions that mechanisms that underlie blast transformation may be potentially different depending on the clonal evolutionary pathway that is used by the transforming clone. So in summary, I just want to acknowledge, the people who have helped out in this particular project. So within John's group and within the MPN program, I really would like to thank John and Vikas Gupta for their support and for moving this project forward. And at OICR, Larry and Phil, who have worked very hard on developing a targeted sequencing panel and getting it to work. And we're hopefully going to be interrogating our samples very soon within the coming weeks. Thank you.